and is a principal scientist from Division of Biochemistry, ICAR-IARI, New Delhi. He was born on 13th August 1973. He did his bachelor's from Punjab University, Chandigarh, master's from Kurukshetra University, Kurukshetra, PhD from same university, Kurukshetra in the field of biochemistry for biotechnology during 2009. He is presently looking after the PG activities as a professor, and his research accomplishments are development of RNA and overexpression constructs for the transformation into soybean for generation of isoflavone defense soybeans, development of cost-effective and eco-friendly protocols for extraction of bioactive phytochemicals like anthocyanins, tocopherols, and isoflavones from soybean, cloning and characterization of antiviral proteins from Bougainvillea into Britannia. And his current research interests are development of high isoflavone soybean using metabolic engineering approach, nanomobilization of enzymes. And he has taught many courses as a course leader and course associate for the PG students of ICAR-IARI, and he has guided 7 MSc students and 3 PhD students. And he has handled more than 12 externally funded projects, and he has got publications of more than 60 in reputed national and international journals, and he has been awarded with and recognized with many awards. To name few, one is ICAR-IARI Best Teacher Award, which is an award which is given for the in the field of best teaching, and Young Scientist Award, Fellowship Award, and he has got other responsibilities like member editorial board, member executive committee of SPPB, and many other. And many of his students has been awarded for their academic excellence like Jawaharlal Nehru Award, ICAR-IARI Merit Medal Award, Inspire Fellowship, Prime Minister Fellowship Award, and Best Poster Awards. So such an eminent person and such a resourceful person is with us, who will be dealing the topic on enzyme kinetics.
Still, I'll take it. So, as has been told by Dr. Sagar, so my main area of concern is the nutritional quality enhancement of the food crops. So, presently I am working on the soybean. So, we are trying to develop the high isoflavone soybeans using metabolic engineering approach. So, we are targeting different enzymes like say isoflavone synthase, then we have another enzyme diagen reductase. So, these are the enzymes which are controlling the accumulation of the isoflavones in the soybean. So, by targeting these enzymes, we are trying to develop the high isoflavone cultivars of the soybean, and that means I have the interest in the various enzymes. But before you start working on the enzymes, you need to understand what these enzymes are, because you need to study the kinetics of these enzymes, you need to study the various parameters which are determining the efficiency of these enzymes. So, before I actually go to the details of the how you actually study the kinetics of the enzymes, I would just like to introduce you with the enzymes, although you all must be knowing about the enzymes. How many of you are from the biochemistry or who have read biochemistry at UG level? All have read. So, that means you have some background about the enzymes. So, what 4 enzymes they are required? So, I will start with the question. Why we want to study the enzymes? Pardon? If you see, for sustenance of any life, you require the energy, and for the production of the energy, you require the food. But food, it will not provide the energy unless until it gets metabolized within the organism. So, that means a metabolism of the various food component, it is going on inside the organism. And how these metabolic reactions they are being catalyzed in the body or in any cell of the organism, it is through the action of the various enzymes. So, enzymes, they are catabolizing as well as the synthesizing various macromolecules present in the cell, and then they are providing the source of the energy. So, this is the importance of the enzymes, and you know that looking at the importance of the enzymes, different fields they are making use of the enzymes. So, let us just start with the just introduction of the enzymes. So, starting from the history of the enzyme, so if you see in the 1700 to 1800, you could find that the digestion of the starch, it was taking place and it was being converted into sugars by the saliva and then by the plant extracts. And then it was also recognized that the meat digestion, it is also possible through the secretions of the various secretion coming from the stomach. And then the fermentation of the sugars to alcohol by yeast, which was studied by the Louis Pasteur in the 19th century, it was also known. But at that time, the enzymes, the things which are actually responsible for these conversions, it was not known. So, the name enzyme, it was introduced in 1878 by the Wilhelm Kuhne, who first used the term enzyme. So, because he studied the fermentation process, and he said that because yeast, they are able to convert the sugars into the ethanol, so that means there is something present in the yeast which is responsible for this conversion. So, all in living, he used the word enzyme, which in Greek means in living. So, he said that something which he called as enzyme because it was living. So, it is responsible for the fermentation of the sugars. And later on, however, it was found that even if you extract the yeast, you take the extract of the yeast, that is also able to ferment the sugars into the alcohol. So, that means the living nature of the organism, it is not required for the fermentation, for the conversion of the sugars into the alcohols. But of course, that term was introduced, so still we are continuing with that term. So, what are the various characteristics of the enzymes which are enabling them to perform such diverse kind of functions, such diverse kind of catalytic activity which they have. So, let us see what are those characteristics. So, as any other catalyst, they are not being consumed during the reaction, because this is the nature of this is the definition of the catalyst, that they are not consumed during the reaction, and same is true for the enzymes. And they are highly specific for the substrate and also for the type of reaction they catalyze. So, specificity, it could be for this substrate which they are acting on, or it could be for the type of reactions which they are catalyzing. So, it could be oxidation, it could be reduction, it could be hydrolysis, it could be isomerization, it could be ligation, it could be lysis. So, they are specific for the reaction which they are catalyzing. So, this is what we mean by the specificity. The specificity does not mean only the substrate specificity, but it also encompasses the type of reaction which is being catalyzed by the enzyme. And of course, the one characteristic which is not present in the various chemical reagents which are used for various reaction, that is the stereospecificity. The stereospecificity, it is specific for the enzymes. So, enzymes, they are able to differentiate between the various stereoisomers, whether they are the cis trans isomers, whether they are DL configuration, whether they are the R and S isomers. The enzymes, they have the capacity to distinguish between these isomers, and this is the property which is very special and specific for the enzymes, which is not possessed by the various chemical reagents, and that enables the enzymes to perform the diverse kind of functions in the cell and as well as outside the cell. And you all must be knowing that most of the enzymes, they are proteinaceous in nature, except of course the ribozymes, which are RNA molecules, but of course their function, it is altogether different. The ribozymes, they are not involved in the metabolism of the macromolecules, but they are controlling the gene expression. So, this is how the ribozymes, they are different from the proteinaceous enzymes. And most of the enzymes, they are globular proteins. Globular proteins, if you recall that in globular structure of the proteins, you have the hydrophilic amino acid present on the surface of the protein, while the hydrophobic amino acids, they are internalized, because hydrophobic amino acids, they tend to avoid the contact with the aqueous environment, right. So, they are globular because they have the hydrophilic amino acid residues present at the surface of the protein. But although this property, it is useful for catalyzing all those reactions which are taking place in the aqueous medium, but these days it has been realized that in industries, various chemical reactions, they make use of the organic phases. So, here it comes as a limitation for the enzymes, because enzymes, they have been evolved over the years to perform their functions in the aqueous environment. But all of a sudden, if you put them in the organic phase, so their efficiency, it goes down. So they are not able to perform their function. So this is a kind of limitation as far as their use in the industries which make use of the organic solvent, it is concerned. And then, of course, you might be knowing they are sensitive to changes in the pH, temperature, and the substrate concentrations. And enzymes, they could be apoenzymes or they could be holoenzymes, because some of the enzymes, they require certain additional factors for performing their functions. It could be organic molecule, it could be metal. So if metal is required, so metalloenzymes are there. If organic molecule, it is required. And it also depends whether organic molecule, it is attached covalently or non-covalently. If it is covalently attached, we call that as the prosthetic group. Otherwise, we will call it as the coenzymes. So combination of the apoenzymes and the cofactor which is required by the enzyme, we call that as the holoenzyme. And then many enzymes, they are multifunctional in nature. That means they are having multiple active sites which are performing the multiple activities. So these enzymes, they are known as the multifunctional enzymes. And then other category of the enzymes which are existing, they are the multi-enzyme complexes. You all know the various metabolic pathways, they make use of the different types of enzymes. But what happens is that sometimes the product of the one enzyme, it gets lost and it is not acted upon by the next enzyme. So that reduces the efficiency of the metabolic pathway. So in order to overcome this problem, what strategies the cell or the nature has devised is that all the enzymes, they come together and they form the multi-enzyme complexes, so that the product of the first enzyme, it never goes in the medium. So directly it is being transferred to the next enzyme, and thereby increasing the efficiency of the whole system. So this is the advantage of having the multi-enzyme complexes, because intermediates, they are never lost in the medium. So because if they come in the medium, so there is a chance of the degradation of those intermediates. So they won't be available for the next enzyme. So this is how the enzymes, they have been classified. The 6 classes are there. You have oxidoreductases, transferases, hydrolases, lyases, isomerases, and the ligases, depending upon the type of reaction which they are catalyzing. But of course, a new classification has come in that a 7th category, it has been introduced recently, I think 3-4 years back. The transporters, they have also been included as a separate class. So these are the various reactions which are being catalyzed by these enzymes, and some of the examples of each category. I won't go into the details of these. This is just for your information. So how the enzymes, they acquire this enormous catalytic power, because they are able to reduce the energy, the activation energy which is required for the reaction. Because what they do is, these enzyme molecules, once they bind with the substrate, what they will do is they will stabilize the transition state. And once this is the energy of the transition state. So what enzymes they are doing is they are having some interactions with the amino acids present at the active sites, and that will reduce the energy of the transition state. So that means the activation energy which was required here, it was more in the absence of the enzyme, but in the presence of the enzyme, you see the amount of the activation energy required, it is very less. So by stabilizing the transition state, these enzyme molecules, they are reducing the activation energy which is required by the enzyme. And once the transition state gets stabilized, so its energy gets lowered, and once the energy gets lowered, so the activation energy required, it reduces, and then a decrease in the activation energy, it enhances the rate of the reaction. So this is how the enzymes step up the rate of the reaction. And the 2 theories, they have been proposed. You all must be knowing that you have loc and key theory, which states that the active site, it is fixed. So once the substrate binds to the active site of the enzyme, and then it gets converted into the product. So there is no conformational change after the binding of the substrate. The structure of the substrate, it is exactly complementary of the active site of the enzyme. But the second theory came, the induced fit theory, which says that the structure of the active site of the enzyme, it may not be exactly complementary to the structure of the substrate. So what happens is the substrate comes, it binds to the active site of the enzyme, it then introduces certain conformational changes in the protein, the enzyme. So that will change the structure of the active site of the enzyme, and it will make it complementary to the structure of the substrate. So this is the difference between the lock and key hypothesis and the induced fit hypothesis. In lock and key, they say that the structures, they are already complementary, but in case of the induced fit theory, they say that after the binding of the substrate, the structure of the active site of the enzyme and the structure of the substrate, they become complementary to each other, and thereby enhancing the binding energy. So this is how the things, they are happening at the active site of the enzyme, because you can have various types of mechanism for conversion of the substrate into the product. You have covalent catalysis, you have catalysis by proximity, you have catalysis by strain, and you have catalysis by acid base reactions. So what actually all these mean? So when coming to the catalysis by proximity, so what enzymes will they will do? They will bring different substrate molecules at the active site of the enzyme. So that means the interaction between, because the concentration of the substrate, it is increasing, so interaction, the chances of interactions, they are also increasing. So they become proximal to each other, and hence the rate of reaction, it gets enhanced. And catalysis by strain, so of course I will explain it in the next slide. So this one. So when, as I said, if you look recall the induced fit theory, what it said, that substrate, it binds to the active site of the enzyme, it introduces certain conformational changes at the active site of the enzyme that will make the structure of the active site complementary to the substrate. But in the process, what will happen is there will be strain on the substrate. The bonds present in the substrate molecule, they will get strained, and a time will come when this substrate molecule, it will be broken into the smaller pieces, and that will be known as the products. So this is what we mean by the catalysis by bond strain, because when conformational changes at the active sites, they are taking place, it is putting a strain on the structure of the substrate, and that in turn is leading to their conversion into the product. So this is the covalent catalysis, because many groups, they are present at the active site of the enzyme, so they are able to form the covalent interactions with the substrate, and thereby facilitating its conversion into the product. So covalent catalysis is also possible, and it is adopted by many enzymes. And acid base catalysis, because if you look at the amino acids which are present at the active sites of the various enzymes, you will recognize that mostly they are the acidic amino acids like say aspartic acid, glutamic acid, or the basic amino acid like lysine, histidine, or the arginine. Of course, the predominant, the most common amino acid present at the active site of the enzymes, it is the serine, right? So either you have the presence of the polar amino acid, or you have the presence of the charged amino acid at the active site of the enzyme. So that facilitates the acid base catalysis, because same amino acid can act as the acceptor of the H ion, or it can act as the donor of the H ion. So that facilitates the acid base catalysis. So this is the catalysis by proximity, which I have already explained, that the concentration of the substrate molecules at the active site of the enzyme, it gets enhanced, so that facilitates their conversion. So now we know what enzymes are, and if we want to estimate the activities of these enzymes in vitro, so what are the tools available with us? So actually, if you see, enzymes, they are not doing anything special, they are converting substrate into the product. So if you want to follow the activity of the enzymes, what you need to do is either you measure the decrease in the concentration of the substrate, or you measure the increase in the concentration of the product. So these are the two alternatives which you have in your hand for the estimation of the activity of the enzyme. But of course, the second one, it is preferred, because it is easier to measure the increase, to go from a lower side to the upper side, but because substrate concentration is decreasing, so initially it is high and it is going down, so it is much less convenient to measure the decrease. So that is why the people, they always prefer to measure the enhancement in the or the increase in the formation of the product rather than the decrease in the substrate concentration. So, but if you want to now follow the increase in the concentration of the product, so what are the tools which you have? So, there should be some property in the product which you should be able to monitor. Only then you can quantify how much product it has been formed, and this property, it could be their ability to absorb certain wavelengths, their ability to fluorescence, give the fluorescence, their ability to give the chemiluminescence, their ability to give certain radioactive reactions. So, depending upon the property which you are measuring for determining the enhancement in the content of the product. So, you have different types of the enzyme assays. So, if you are measuring the increase in the absorbance or decrease in the absorbance, those methods, they will be known as the spectrophotometric methods, and other, if you are following the radioactivity, so radio assays, they are available. If you are monitoring the fluorescence, so fluorescent assays, they are there. So, the point which I want to convey here is that you need to identify a special property of the product which you can monitor during the course of the reaction, and depending upon that you have to choose your assay method. So, I have given few examples here, so the direct UV light, the radio chemicals, or the induced UV light, and of course, yeah, the methods which are usually employed, they could be continuous method or the discontinuous method. So, anybody aware about it, what these two are, continuous and discontinuous assay methods for the assay of the enzymes? In continuous assay, what we do is we mix the enzyme, substrate, and the other requisite material for the reaction mixture, and then we put it for incubation for a certain period of time, and after the end of the incubation period you measure the change in the concentration of the product or the decrease in the concentration of the substrate, right. So, this is a continuous method, but in discontinuous method what we do is, so after every interval, say 30-second interval, we are measuring how much increase it is taking place, right. So, you are following the course of the reaction during the whole incubation period. So, it is not that only after the just like say a normal PCR and the RT-PCR, you had a lecture just now. In PCR what we are doing is, after the end of the say given number of cycles you are measuring the product, but in RT-PCR what you are doing is you are following the progress of the reaction in the real time. So, same thing in case of enzymes as well, that if you are taking the OD at certain intervals, time intervals, so you are following the course of the reaction rather than measuring the end product in the end. And this is how you express the activity of the enzyme. So, 1 unit of enzyme, it is defined as the activity required to convert 1 micromole of the substrate into product per minute. But you also have a higher unit of the enzyme activity. What is that unit known as? This is the international unit. The other unit, it is the Catal. The Catal, it says that amount of the enzyme which converts 1 mole of the substrate into product per second. So, this is high unit. The international unit is the micromole of the substrate getting converted into product per minute. Time is also higher, the amount is less, but in case of Catal, amount is more. In place of micromole you have mole, number of moles of substrate getting converted into product per second, time is also less. So, it is a higher unit of the enzyme activity, but the most commonly used one, it is the international unit. There was a scientist by the name Arthur Kornberg. You all must be knowing what is the contribution of the Arthur Kornberg. DNA polymerase, yeah, very right. He worked extensively on the DNA polymerase and determined its role in the DNA replication. And after working for such a long period on the enzymes, so he gave 10 commandments for the enzymes, and you can see here that out of the 10 commandments, the 4 one which I have written here in the red, they are really very important if you want to start the work on the enzymes. The first one you can see here, the not waste, clean thinking on the dirty enzymes. So that means if you want to study the enzyme kinetics, so you need to have those enzymes in the purified form, otherwise the results which you will be getting, they will not be reproducible. So that's why he said that don't waste clean thinking on the dirty enzymes, because whatever howsoever good idea you might be having, but that is of no use if you are working with the dirty enzyme. So purification of the enzyme, it is a must. And then note waste clean enzymes on the dirty substrate. So that means the purity of the substrate, it is also equally important. Not only the purity of the enzyme, but purity of the substrate, it is also. This is a problem which Kornberg faced, because he was not able to explain the unwinding of the DNA, how the DNA replication it is actually starting, which is the primer molecule. And the reason was that he was using the impure DNA preparation, and when he replaced the impure DNA preparation with the pure preparation, he could very easily explain how actually the replication process it is taking place. And this forced him to state this, that no waste clean enzymes on the dirty substrate. And be aware that the cells are molecularly crowded. So what does it mean? Molecular crowding. So now what we are doing is we extract the enzymes, put it in the buffer, and what is the concentration of the enzyme or the other molecules in that extraction buffer? It is very, very low. But imagine the milieu or environment in the cell. In the cell you have thousands of the proteins, thousands of the lipid molecules, thousands of the other macromolecules, they are present. So that means there will be lot of crowding in the cell, and which you are not simulating when you are doing the assay of the enzyme in the vitro. In vitro you are using very dilute conditions, and we are fortunate that we are able to get the activity of the enzymes even in the dilutes after say 1000 to 100 times of dilution. But in the cell, the environment, it is altogether different. It is highly concentrated. I won't use the word concentrated, because concentrated word we use when we are using same solute. Crowding we use when we have different types of solute. So that's why it is not the high concentration, it is the molecular crowding. So we need to make corrections with regard to the molecular crowding. Although we can use the dilute preparations for the assay of the enzymes, but we need to extrapolate the things taking molecular crowding into the consideration, and employ enzymes as unique reagents, because nowadays everybody is after the use of the organic solvents, and you all know the consequences of the use of the organic solvents. So they do have the harmful effects on the not only on the human beings, but also on the growth and development of the plants if they come in the soil as well. So that means you need to have reagents which are eco-friendly, which are green. And that's why I said in the beginning that you need to have enzymes which can work in the hydrophobic environment as well, because by their intrinsic nature they are able to act only in the aqueous environment because of their water solubility, because of their hydrophilic nature, and because of their globular nature, which means that they have the hydrophilic groups at the surface and not the hydrophobic one. So as I said that it is of utmost importance that we use the enzymes in their purified form. So how to go about it? So you have different conventional biochemical techniques which can be used for the purification of the enzymes. You can either use the first centrifugation can be employed, then chromatography can be employed, then electrophoresis. I won't go into the details of the technique because time will not permit me to do so. But in some other lectures we can discuss about the techniques, how they can be employed for the purification of the proteins or the enzyme per se. So you can use chromatography, you can use centrifugation, you can use the electrophoresis, and you have different types of chromatographies. You can either use the gel exclusion chromatography, you can use ion exchange chromatography, you can use affinity chromatography for the purification of the enzymes. And once you purify, you need to see how much purification you have obtained. The fold purification, it is very, very important, because it will tell you when you can make it for the downstream purposes. And now coming to why we need to study the enzyme kinetics, because we have understood about the enzymes, we have understood about the need for the purification, but where these purified enzymes now they will be employed for studying the kinetic properties. What is the need for studying the kinetic properties? Because everybody wants that they should have the enzymes which can work very fast. And if you really want to develop such kind of enzymes, you first should know that what are the parameters contributing towards the catalytic efficiency of the various enzymes, and from that you will come to know about that parameters responsible for the catalytic efficiency of the enzymes. So that will come through enzyme kinetic studies. So there are certain parameters known as the K catalytic, Km, Vmax. So I will come to the details of those parameters. So how they are actually related with the efficiency of the enzyme. And then we need to optimize the enzyme activity, because as I said, the enzymes, they are highly susceptible to changes in the pH, temperature, or the substrate concentration. So what are the optimal conditions required for the activity of the enzyme? So that you will come to know through the enzyme kinetic studies. And then if you want to study the role of enzymes in the metabolism, because I will come to the details of those, how these enzymes they get regulated, so then again you need to study the kinetics of those enzymes. And of course the drug development also, because you might be knowing that most of the drugs which are there in the market, so they are actually targeting one or the other enzyme. For example, the statin, which is being used for controlling the blood cholesterol level. So actually this is the inhibitor of the HMG-CoA reductase enzyme. So for every drug if you take, even the drug which I am taking, that is for controlling the blood pressure, it is also an inhibitor of one enzyme, right? So most of the drugs available in the market, so they are actually the inhibitor of one or the other enzyme. So we need to identify the targets in order to in order to move forward as far as the drug development it is concerned. So the Michaelis-Menten equation, this is the basis of the enzyme kinetic studies. So you can see here that enzyme, it binds to the substrate, and K1 it is the binding constant for binding of the two, and it forms the ES, the enzyme substrate complex it is formed. And then this enzyme substrate complex, it gets broken into the free enzyme, and then you get the final product. And these are the constant, dissociation constant for the reverse reactions. So because we don't have that much time that I can tell you how this equation it is derived, but for the time being you just remember that after taking into the consideration the rate of appearance of the ES complex and the rate of disappearance of the ES complex. So you come to an equation that is known as the Michaelis-Menten equation, and this Michaelis-Menten equation, it has this form. It includes the Vmax, that is the maximum velocity which can be achieved by the enzyme, and then it includes a parameter known as the Km, that is known as the Michaelis-Menten constant. So these are two very, very important constants, and they carry great deal of significance. So I will tell you and what these constants they are, and of course the V0 it is the initial velocity. So here I would again like to pinpoint that whenever you are doing the enzyme kinetic studies, so always take the study the reactions during the initial period only, because initial period, when I say, what do I mean is that when say you have started with the 100 millimolar of the substrate, so till the time 20% of the substrate it is consumed, so that is your initial velocity, because after that the changes in the substrate concentration it will influence the activity of the enzyme, because as I said in the beginning that the activity of the enzyme it is dependent on the concentration of the substrate. So usually when we are doing the enzyme assays, we are taking substrate concentration in excess of the enzyme. So it is very, very large. So in the initial velocity period, there won't be any significant change in the concentration of the substrate, and that's why it won't have the influence on the activity of the enzyme. So whatever influence you are getting, that is because of the intrinsic nature of the enzyme. So this is what I mean by that you need to study the enzymes during the initial velocity period, when the substrate concentration, it is 20% of the initial substrate concentration taken during the assay of the enzyme. So this is how the graph of the substrate concentration against the velocity looks like. You can see that this graph, it never touches the Vmax level. Why is it so? Why it does not touch the Vmax? Because at higher, somebody was, substrate will be converted, but substrate we are taking in large excess, all the substrate molecule, because if you are taking substrate concentration in excess, what will happen is the enzyme, it will get saturated with the substrate. So you don't have the leftover enzyme for conversion of the substrate into the product. So when that situation comes, so howsoever increase of the substrate concentration you may do, but it will not lead to the enhancement in the rate of the reaction. So it will never touch the Vmax. And how do we calculate the Km, the Michaelis-Menten constant? The Km, it is calculated as it is defined as the substrate concentration required to achieve the half of the Vmax. So this is the Vmax, and half of the Vmax is this. So corresponding to this, the amount of the substrate which is required, that is known as the Km. And Km, as I said, it is very, very important kinetic constant, because it tells you about the affinity of the enzyme for its substrate. So a enzyme might be acting on different types of substrates, but it will have different Km value for the each substrate. So in the cell you know that say 10 substrates are there for a particular enzyme. So on which substrate the enzyme will act out of those 10. So it will depend upon the Km value of the enzyme for the each substrate. So it has a inverse relationship. The higher the Km value, lower is the affinity of the enzyme for the substrate, and lower is the Km value, higher is the affinity of the enzyme for the substrate. So this is the point which I want you to remember for your whole life, that Km, it has the inverse relationship with the affinity of the enzyme for the substrate. So higher the Km, lower is the affinity, lower the Km, higher is the affinity. So now you can very easily make out how the enzymes are making decision while working in the cell. So as I said, if there are 10 substrates, out of those only a few will be selected depending upon the one which are having the low Km value, that means the high affinity. So this is what you can conclude from this graph, that if say substrate concentration it is equal to Km, so we can see here that from the Michaelis-Menten equation you can see the Vmax is half. So that means it is Km value, and then when S is more than Km, so what is happening is V is directly proportional to V is almost equal, not directly proportional, V is equal to Vmax, and when S is less than Km value, V is directly proportional to S. So that's why in the previous graph you can see here, when substrate concentration is less, so you have large increase in the rate of the reaction, but when you have intermediate it is lesser increase, but at the top you don't have much of an increase. So we call this type of graph as the hyperbola. So in those enzymes which are following the Michaelis-Menten kinetics, so they will give you a hyperbola graph for the velocity to substrate. So as I said, the Km, it is a measure of the affinity of an enzyme for its substrate, and this is the Km value for different kinds of the substrate. And as you can see, the hexokinase, it can act on these 3 substrates, but for each substrate it has the different value of the Km. And then if you know the Km value of the enzyme for its substrate, so then it is very easy to know how much substrate concentration it is to be used during the reaction mixture, because otherwise it will be arbitrary. So you will be just making the guesses how much substrate needs to be added into the reaction mixture. But if you have the you have calculated the Km value, you know that how much is the substrate required for the reaction to take place. And this is Km is K minus 1 K2 divided by K1. So if you recall, in the previous slide I mentioned these constant, the K2 is the conversion of the ES into the product, K1 it is the binding of the E plus S into ES, and K minus 1 it is the dissociation constant for conversion of ES into the E plus S. So this is how you calculate the Km. And then another parameter which is of utmost importance as far as the enzyme kinetics it is concerned, it is the turnover number. What do you understand by it? Although it is written in the slide, but still, what do you understand by turnover number? Number of molecules of the substrate getting converted into product per unit time per molecule. This is important because many times if you are given the concentration of the enzyme, so concentration it is in the millimoles or the micromoles. So you are considering that you are calculating the turnover number in terms of number of moles, but it is actually not so. So you have to calculate per molecule of the enzyme. If you want to convert mole into the molecules, what is the relation? Avogadro number of molecules. So 1 mole of the substance or the enzyme, it will contain 6.02 into 10 to the power 23 molecules, that is the Avogadro number. So from this relationship, first you have to calculate how many molecules of the enzymes they are present in your reaction mixture, and then you can calculate the turnover number by taking the definition as the number of substrate molecule getting converted into product per unit time per molecule, and it is usually represented as K catalytic. Okay. If there are two steps only, the E combining with the S giving you ES and then ES getting converted into product, then I wrote, let me go back, this K2. So if there are only two steps in a enzymatic reaction, then this K2 it is actually the K catalytic. But there are many reactions where you have multiple steps. So after say 3 or 4 steps you will get the formation of the product. So then the K catalytic it will be different, it will be sum of the rate constant of the each and every step. But for this one the K2 is equally good as the K catalytic. So and this is the turnover number of the various enzymes, and you can see that some enzymes like catalase and the carbonic hydrase, they are really very efficient. So see the number of molecules of the substrate getting converted into product per second per second or per minute, the number of molecules, it is more than say 400 lakhs, right. So they are really, these enzymes they are known as the catalytically perfect enzymes. So I will tell you how we determine the catalytic perfection of the enzymes. And how do you calculate the value of the Km and Vmax? Because as I said that you are not able to calculate the value of the Vmax using the V versus S curve, because the curve never reaches the Vmax. So you cannot calculate the Vmax from that curve, and if you cannot calculate the Vmax, you cannot calculate the Km value, because Km value is nothing but the substrate concentration required to achieve the half of the Vmax, right. So that means Km and Vmax they cannot be derived from the V versus S plot. So you require the other means and ways to have the calculation about the Km and Vmax, and Lineweaver-Burke plot, it is one of the most common plot which is being used for the calculation of the Km and Vmax. And here we are just rearranging the Michaelis-Menten equation. It is no, we are not doing something special. So we just start from, say this is the Michaelis-Menten equation, you take the inverse of Michaelis-Menten equation and then you can rearrange it, and you will find that there are two reciprocals, 1 by V and 1 by S, and you plot 1 by V against the 1 by S. So that is why this plot is also known as the double reciprocal plot, because you are plotting the two reciprocals, 1 by V against the 1 by S. And if you see this equation, it is a straight line equation, y is equal to mx plus c. So this will be the y intercept and this will be the slope. So that is why here in this you can see this is the slope and this is the y intercept, and from this you can also calculate the x intercept, that is minus 1 by Km. So you remember this because this will be very useful when we will study about the inhibition. So in Lineweaver-Burke plot, enzymes which are following the Michaelis-Menten equation, the slope is Km by Vmax, y intercept is 1 by Vmax, and x intercept it is minus 1 by Km. And of course there are certain limitations also for the Lineweaver-Burke plot, because when S is very low, that means 1 by S is very high. So but if you want to follow the reaction here, so at very high substrate concentration it is it will never meet the x axis. You have to plot it on a very large graph paper, only then you can get the union about of the plot with the x intercept. So that is why sometimes people they do not prefer to use the Lineweaver-Burke plot. Of course this is still out of the available methods, this is still the most commonly used method, but other methods I will just want to acquaint you with. The Addy-Hofstee is the other one, the Hannes plot, and all these plots they have been obtained by rearranging the Michaelis-Menten equation in one way or the other way. So in Lineweaver-Burke plot we have taken the inverse. So in these plots what we are doing is we are taking the inverse and then multiplying it with the certain factor and then rearranging it. And then we are in Addy-Hofstee V by S is plotted against the V. In case of Hannes plot, S by V is plotted against the S, and then there is a direct linear plot. So this is something different, because here you are plotting the things which we actually want to calculate. If you see here, the Vmax is plotted against the Km. So in direct plot, the Vmax is plotted against the Km. I won't go into detail because it will be confusing for you. Of course, we are discussing it in detail to the biochemistry students, but you should be knowing that apart from the Lineweaver-Burke plot, you have other plots available for the calculation of the Km and Vmax. These are the Addy-Hofstee plot, you have Hannes plot, you have the direct plot. So this is what I was talking about, that some enzymes they are catalytically perfect enzymes, and when we talk about the catalytic perfection of the enzyme, so you need to measure a kinetic parameter known as the specificity constant, so which is nothing but the ratio of the K catalytic by Km. So just understand it, when I say that K catalytic by Km, it is the measure of the perfection of the enzyme. When K catalytic by Km is high, what actually it means? It means that K cat is very high and Km is low. If K cat is high, that means more number of substrate molecules they are getting converted into the product, right? And when Km is low, that means more number of substrate molecules they are binding to the enzyme, because I told you there is a inverse relationship between the Km and the substrate affinity of the enzyme for the substrate. If Km is low, more of the substrate it will bind to the enzyme and more of it will be converted into the product, and that is why the K catalytic by Km it is considered as the measure of the perfection of the enzymes. So higher the K catalytic by Km, more is the catalytic perfection of the enzyme. So these are the few enzymes which are considered to be catalytically perfect, and there is a limit to it, because you see the enzyme catalyzed reaction, it is just like any other reaction. It all boils down to how many collisions they are taking place between the substrate molecules and the enzyme molecules, and whether these collisions they are effective or not. So if they are in perfect orientation, then they will be effective, and if orientation is improper, then it will not be effective, right? So that is why I said there is a limit to perfection of the enzyme, and the maximum limit is which is controlled by the diffusion. So if you have more diffusion of the enzyme and the substrate, so naturally more collisions will take place, and then naturally more substrate molecule will be getting converted into the product. And 10 raised to power 9 it is the limit which is set by the diffusion of the various molecules in a system, right? So maximum value of the K catalytic by Km it could reach to the diffusion control limit, that is the 10 raised to power 9. So if any enzyme it is having say K catalytic by Km in the range of say 10 raised to power 6 to 10 raised to power 9, usually 10 raised to power 9 it never achieves, up to 10 raised to power 8 if it is. So, we consider it as the catalytically perfect enzyme. So, now another, once you know about the K catalytic, Km and the Vmax, so another tools which is available in the hands of the enzymologist, it is the inhibition of the enzyme, because you can exploit it for your own use if you know how to inhibit the particular enzyme, because people as I was discussing that most of the drugs they are being developed by the inhibition of the various enzymes. So, it is very important in how to have the maximum inhibition of the enzyme. So, what are the things which are need to be taken into consideration. So, if you see there are two types of inhibition, reversible and irreversible. As the name suggests, the reversible it can be reversed, and irreversible because there is a covalent linkage between the inhibitor and the enzyme, so it cannot be reversed. So, this kind of inhibition it is known as the irreversible inhibition, and reversible it is again of 4 types. You can have competitive, uncompetitive, non-competitive and the mixed type of the inhibition. So, let us see what these different types of inhibitions are. Coming first to the competitive inhibition. So, the beauty of this competitive inhibitor is that it only binds to the free enzyme. So, this is the normal reaction, E binds to substrate, ES is formed and ES getting converted into the product. So, inhibitor because it is having the structure similarity with the substrate. So, that is why the name is competitive inhibition. That means there is a because of the structure similarity there is a competition between the substrate and the inhibitor for binding at the active site of the enzyme. That is why this is known as the competitive inhibition, and now the battle it will be won by that molecule which is having the high concentration. So, if substrate concentration is high, more binding of the substrate will take place on the active site of the enzyme. If inhibitor concentration is more, so more binding of the inhibitor will take place on the active site and there will be more inhibition. And this is reversible inhibition. Why? Because both substrate and the inhibitor they are binding to the same site. So, if you enhance the concentration of the inhibitor, so it will replace the substrate, and if you enhance the concentration of the substrate it will replace the inhibitor, and once the inhibitor it is replaced, that means the inhibition it has been reversed. So, that is why this inhibition it is known as the competitive inhibition. But remember that inhibitor is able to bind only to the free enzyme, not to the enzyme substrate complex, and these are the changes of course. When I say that this can be reversed by enhancing the concentration of the substrate. So, what do I mean? What should be the effect on the Km value by taking this point into consideration, that if you add more of the substrate, so then inhibition can be reversed. Km value it will get enhanced. The Km value is the substrate concentration required to have half of the Vmax. So, that means now in the presence of the inhibitor you require more and more substrate concentration to achieve half of the Vmax, and if you require more substrate concentration, that means the Km value it will get enhanced. And of course, these are lengthy equations, we cannot derive it here, but in nutshell you will see that this Km value it has been enhanced by a factor of 1 plus I by Ki, where I is the inhibitor concentration and Ki is the inhibitor constant. So, we can determine the value of the inhibitor constant as well. We have different types of plots available, I will not go into the details. Dixon plot is there, then you can plot certain secondary plots that will give you the value of this inhibitor constant. So, but in nutshell, as a result of the competitive inhibition, what will happen, the Km value it will gets enhanced, and what will be the result of this if you plot the Lineweaver Burke plot. So, as I said that please remember the value of the slope, y intercept and the x intercept. The value of the slope was Km by Vmax, y intercept was 1 by Vmax, x intercept was minus 1 by Km. Now, since Km it has been affected, so what which two parameters will be affected in the line wave bouquet plot, x, y or slope, x intercept, y intercept or the slope, because slope has Km value in it, so the slope will be affected. It will increase or decrease, slope will increase or decrease. Slope is Km by Vmax, Km is increasing, so that means Km by Vmax will increase, so the slope it will enhance. And what will be effect on the y intercept? Y intercept does not have any component of the Km, it is only 1 by Vmax, so it will not be affected. And x intercept, because it is having the Km component, so it will be Km, it will increase, so 1 by Km it will decrease. So that is, yeah. Pardon? No, this is the definition. Vmax will not change because Vmax is never achieved. As I said, the Km, it is just the definition of the Km, that amount of the substrate required to achieve the Vmax. Vmax is already there, right? I will tell you, because in the next type of inhibition the Vmax will change. I will tell you how the this Vmax gets changed. So is it clear? So you can see that in the absence of the inhibitor, this was the curve. In the presence of the inhibitor, the slope it has been increased, the y intercept it remains same, while the x intercept it decreases. So the reason behind this is the change in the value of the Km. Is it clear? And why Km changes? Because you require more substrate concentration to get Vmax in the presence of the inhibitor. So next type of inhibition, it is the uncompetitive inhibition. This is again a unique type of inhibition. Before I go to the uncompetitive, I will just give you one example of the competitive inhibition. So the classical example, it is of the inhibition of the succinate dehydrogenase enzyme by the malonate. Malonate, if you see the structure, it is very similar to the succinate. So it could act as a competitive inhibitor of the succinated dehydrogenase enzyme. So that is the classical example of the competitive inhibition. So again, coming to the uncompetitive inhibition, as I said, it is a unique type of inhibition because the inhibitor, it is not binding to the enzyme, free enzyme, it only binds to the enzyme substrate complex. As you can see here, it is binding to the enzyme substrate complex. So that means the binding of the substrate to the enzyme, it is causing a conformational change in the enzyme, and that is exposing the binding site for the inhibitor. So once the substrate binds to the enzyme, the binding site for the inhibitor, it is exposed, and that is why you can get the formation of the enzyme substrate inhibitor ternary complex. But unfortunately, this ternary complex, it is a dead end complex. It cannot result in the formation of the product, and that is why the Vmax it decreases, because Vmax it is dependent on how many enzyme molecules they are bound to the substrate, that means the concentration of the ES, right. So when inhibitor binds to the ES, what is happening is some of the ES it is getting converted into dead end complex ESI. So that means less concentration of the ES it is available for conversion into the product, and that is why the Vmax it gets reduced, because Vmax is nothing but K2 into ES or K catalytic into ES, concentration of the enzyme substrate complex. So when enzyme substrate complex gets reduced, so that means the Vmax it will also get reduced. And what will happen to the Km value in this case? Km value, it will increase, decrease or remain constant? Actually the Km value it also gets decreased in the presence of the uncompetitive inhibitor, and the reason behind this is that as more of ES gets converted into ESI, so cell wants to form more of the ES, right. The enzyme forms to act bind with more of the S to form the ES. So this is you must have read about the Lee-Chatelier principle if you at UG level or maybe at 12th standard. So, they discuss the Lee-Chatelier principle is that if you remove the product from the reaction, so then more and more of the product will form. So, that is why those reactions in which one of the product is gaseous in nature, it gets removed from the system. So, they have more rate of the reaction. So, more of the ES will try to form, and it will appear that more substrate is binding with the enzyme. So, it will appear that more the Km value it has been reduced, right. So, the Km apparent it is lesser, and Vmax it is also reduced in the presence of the uncompetitive inhibitor. And now you will explain why this kind of curve it is obtained. So, when Km is getting reduced, Vmax is getting reduced by the same amount, what will be the effect on the slope? Slope is nothing Km by Vmax, both Km and Vmax they are getting reduced by the same amount. So, net values it will remain same. So, that is why you are getting the parallel lines. And what will happen to the y-intercept? Y-intercept is 1 by Vmax, Vmax is decreasing, 1 by Vmax it will increase. So, that is why in the presence of the inhibitor y-intercept it has increased. What will happen to the Km, the x-intercept? Because Km is decreasing, 1 by Km it will increase. So, that is why in the presence of the inhibitor 1 by Km it has increased. So, that is why you are getting the parallel lines, because both Km and the Vmax they get reduced by the equal amount, and that is why the slope it remains constant, x-intercept it increases, y-intercept also increases. So, these are the things which you need to take into account when you are doing the enzyme kinetic studies. So, what you are doing in reaction mixture, you are taking different substrate concentration, you are getting different rates of the reactions, then you are plotting 1 by V against the 1 by S. Then again doing the same experiment in the presence of the inhibitor, see what kind of graphs you are getting, and on the basis of those graphs you can tell whether it is competitive inhibition, uncompetitive inhibition, and then you can decide which type of inhibitor it has to be used, because uncompetitive inhibition inhibitors they are usually used when you have very high substrate concentration in the cell. When you have very high substrate concentration in the cell, what will happen that most of the enzyme it will be present in the bound form. So, there the competitive inhibitors they will not work, because competitive inhibitors they bind only to the free enzyme, but if you have high substrate concentration, so most of the enzyme it will be present in the ES form. So, there you require the uncompetitive inhibitor, because they have the ability to bind with the bound form of the enzyme. So, this is what we have discussed. It lowers the y-intercept increases, x-intercept it is increase here, not the decrease, and Km it will remain same. So, coming to the non-competitive inhibitor. So, non-competitive inhibitor it is a mix of these two. So, inhibitor it has the ability to bind with the free enzyme, it has the ability to bind with the enzyme substrate complex as well, and even the substrate can also bind to the enzyme inhibitor complex, and thereby leading to the formation of the enzyme substrate inhibitor ternary complex, which is a dead end complex, which will not result in the formation of the product. So, what should be the effect on the Km and Vmax in case of non-competitive inhibitors? What should happen to the Vmax? You know the basis how the things they are calculated. Vmax is dependent on the concentration of the ES, right. So, what is happening to the concentration of the ES? It is getting reduced, because some of it is getting converted into the dead end complex. So, Vmax it will get reduced, and what will happen to the Km in case of non-competitive inhibition? Pardon? It will remain same. Why? How do you define the Km value? amount of the substrate concentration required to achieve the value Vmax, half of the Vmax, right. So, then what should be the effect on the Km value in this case? Is the binding of the substrate, because Vmax it will depend upon the binding of the substrate to the enzyme. So, binding of the substrate, do you think it is affected in any way? Because here the binding of the inhibitor and the binding of the substrate, they are independent of each other. So, substrate is binding to other side and inhibitor is binding to the another side. In case of competitive inhibition, both they were binding to the active site of the enzyme, but here in non-competitive inhibitor, both are binding to the exclusive sites. So, they are not interfering the binding of each other. So, that is why there is no point of the binding of the substrate being hindered by the presence of the inhibitor. So, that is why the Km value it remains same in case of non-competitive inhibition, only the Vmax value it will be reduced. And what will the effect on the various parameters? The slope, which is Km by Vmax, Vmax is changing, it is decreasing, so slope it will increase. You were decreasing it, you were saying like this. So, it because slope is Km by Vmax, Vmax is denominator is decreasing. So, that means overall quantity it will increase. So, there will be increase in the slope. And what about y-intercept? Because y-intercept is 1 by Vmax, so Vmax is decreasing, 1 by Vmax it will increase. So, this y-intercept will increase, and x-intercept it will, because Km is not changing, so x-intercept it will remain same. So, this is how you can distinguish the three different types of inhibitors from each other based on the Lineweaver-Burke plot obtained in the absence and in the presence of the inhibitor. So, this is the mixed type of inhibitions. Again, this is a very complicated kind of thing, so I will not go into the details. Because mixed inhibition, it could be competitive, non-competitive, it could be non-competitive, uncompetitive, so different types of inhibition. But you should know that if you are performing the experiment in the lab, sometimes you can get this kind of graph as well. So, it is not coming as per your expectations that the x-intercept is changing, y-intercept is changing, or slope is changing. So, it is not falling in either of the categories. So, it is somewhat like this, the graphs they are meeting either above the x-axis, or sometimes they will also meet below the x-axis, right. So, this is an indication that it is a case of mixed inhibition, it is not falling in any of the three categories. So, then as I said that enzyme inhibition it could be irreversible. When it is irreversible? When inhibitor forms a covalent bond with the active site of the enzyme. So, that kind of inhibition then cannot be reversed by changing the concentration of the substrate. So, this kind of inhibition it is known as the irreversible inhibition, and the classical example it is the inhibition of the chymotrypsin with the diisopropyl fluorophosphate. You all must be, if we are doing the enzyme assays, so always we use the inhibitor of the proteases during the reaction mixture, so that our protein it remains intact. So, that is why these inhibitors they are very, very important, because chymotrypsin it is nothing but a protease. So, you need certain inhibitors to check the activity of those protease enzymes, and DFP is one of them, and it is fortunately a irreversible inhibitor because it forms the covalent linkage. So, this is the linkage which is formed, HF it is released, and then this molecule get covalently attached, and you will get these kinds of curves when you have the irreversible inhibition. So, then sometimes you will find that at higher substrate concentration your enzyme gets inhibited, because this is having a great significance, because many times in the cell decision has to be made whether there is a requirement for a particular reaction to take place or not. And if there is no requirement, say ATP synthesis, if ATP is already available and the cell wants to decide whether ATP should be synthesized or not, so then sometimes your substrate it will also inhibit the enzyme. So, that kind of inhibition it is known as the substrate inhibition. and the very good example is of the succinate dehydrogenase enzyme. So, if you get the higher substrate concentration or higher concentration of the succinate, so what happens is in normal low substrate concentration, this is how this succinate it will bind to the enzyme, and then a double bond will be introduced here and you will get the formation of the fumarate. So, this is a reaction catalyzed by the succinate dehydrogenase enzyme. Succinate gets converted into fumarate, right. But if you have higher substrate concentration of the succinate, so what will happen? The binding will take place like this. One succinate molecule it will bind this way, and before this carboxy binds here, the second molecule of the succinate comes and it binds here. So, now the enzyme is nowhere. So, it cannot catalyze the reaction, it cannot incorporate the double bond between the two methylene groups, because they are not exactly positioned in that way. So, you will not get the formation of the fumarate. So, we say that higher concentration of the succinate, it has inhibited the enzyme, and this is known as the substrate inhibition, when the substrate itself is inhibiting the enzyme. And you will get this kind of curves, because normally you see in V versus S curve, the curve goes like this and then a plateau comes. But here what is happening is at higher substrate concentration, the velocity it is getting decreased. So, if you are getting this kind of curve, so that is an indication that there is a substrate inhibition, otherwise a plateau will come. But if a dip comes, so at higher substrate concentration, so it's a clear cut indication that substrate inhibition has taken place. And similarly, if you plot 1 by V against the 1 by S, so before it reaches the y axis, so it will start going parallel to the y axis. So, this is again an indication that substrate inhibition has taken place. So, these are the enzymes which are following the Michaelis-Menten equation, but there are certain enzymes, especially the enzymes involved in the regulation of the metabolic pathways. We know in every metabolic pathways there are certain regulatory enzymes, and most of these regulatory enzymes, they are actually not following the Michaelis-Menten equation. They are having a different kind of kinetics. These enzymes, they are known as the allosteric enzymes, because the kind of curve V versus S curve which you are getting, it is not hyperbola, you are getting the sigmoidal curve. And the beauty of this sigmoidal curve is that till a particular concentration of the substrate, there will not be much effect on the velocity, but a threshold will come. If you go beyond that threshold, a slight increase in the substrate concentration, it will shoot up the rate of the reaction, and this is what is required during the metabolism in the cell, that you want to change a slight concentration of the substrate, and that should change the activity of the enzyme drastically. And that is why the most of the regulatory enzyme you will find that they are the allosteric enzymes. And another beauty of these allosteric enzymes is that they show the phenomenon of cooperativity. Cooperativity, of course, I haven't taken the slide here. The cooperativity actually means that when the binding of the one substrate molecule, it facilitates the binding of the other substrate molecules, and very good example is of the hemoglobin. The hemoglobin, it uses the oxygen as the substrate. So, binding of the one oxygen molecule, it will facilitate the binding of the other oxygen molecule, and this is true for most of the allosteric enzymes, that they facilitate the binding of the one substrate molecule, facilitate the binding of the other, and that is why they are the regulatory enzymes. Because if such kind of phenomenon it is observed, you will be surprised to know that if from 10% of the enzyme activity, if you want to go to the 90% of the activity, you require change in only 9-fold change on the substrate concentration is required. But if you want to achieve the same thing in the Michaelis-Menten enzymes, you need to enhance the concentration of the substrate 89 times. So, this is the difference between the enzymes following the Michaelis-Menten kinetics and the one which are following the allosteric phenomenon. So, you require very small change in the substrate concentration to get the desired effect. And here we do not use the word Km, we use the K.5, that means the meaning is same, but because they are following altogether different kinetics, so we use K 0.5. So now you know what enzymes are, how they are working, and you know the means to use them for and kinetic studies. You first need to purify those enzymes, and only then you should study their kinetics, and then how these enzyme kinetic studies they could be applied. And you all know the examples of the I think it is. Yeah. So as I told you that most of the enzymes inhibitors they have been used in the drug development, and the classical example in the agriculture field of the glufosinate inhibition. Which enzyme it targets? You all must be knowing it from the agriculture. You heard about Bilaphos, Basta, phosphenothryacin? Never heard about these herbicides?
Never studied the mechanism of action.
Yeah. So actually these glufosinate, it is a herbicide which targets the glutamine synthetase enzyme. So there is accumulation of the glutamine synthetase enzyme, it converts glutamate into glutamine. So if that enzyme gets inhibited, so there will be accumulation of the glutamate, and ammonia it will get accumulated, and you know if ammonia gets accumulated it will have the toxic effect that will lead to the death. And similarly glyphosate inhibition, which is the target? Pardon? As agriculture student it is not expected. These are the questions which are usually asked during the competitive examinations. The EPSPS, enoyl pyruvyl shikimate phosphate synthase, the enzyme which is involved in the synthesis of the aromatic amino acids. So and then inhibition of the acetyl CoA carboxylase, you know the cyclohexane dione and alloxyphenoxypropanate. So they are targeting the enzyme acetyl CoA carboxylase, which is involved in the synthesis of the fatty acids. So all these inhibitors they have been used for controlling the one pathway or the other in order to get the desired effect. And the recent application which is coming is the enzyme inhibition based biosensors. So these days you all know that so much importance it is being given to testing of the foods for various pesticides. So if you want to do that, how you will do? Here the enzyme inhibitor based studies they can be useful. You see this is the enzyme which has been immobilized, and this convert this substrate into the product. But if your food contains the pesticides, and these pesticides they are the known inhibitor of the immobilized enzyme, what will happen? The inhibitor it will bind here, the pesticide it will bind to this enzyme, so it will inhibit it. The substrate it will not be converted into the product. So if when you are adding your analyte or the sample on this immobilized enzyme, and after that you are measuring the activity of the enzyme, if you are getting the decreased activity or you are getting no activity, so that is an indication that in your sample the pesticide it was there which was inhibiting that enzyme. So such kind of biosensors now they are being developed. You all know the biosensor for the glucose, because in most of the families the glucose estimation it is done by various kits, glucometers are there. So what is the principle involved there? Which enzyme it has been immobilized there? No, amylase. Glucose oxidase enzyme it has been immobilized on that, right? So then you can follow the whether the kinetics of that. So, similarly here in this one also you need to identify certain enzymes which can be immobilized. The common one, they are the tyrosinase enzyme, it is very commonly used in such kind of biosensors, and you need to first study the pesticide which you are going to analyze, what is the target of that pesticide out of the available enzymes, and then from that you can choose a suitable target and then you can immobilize on the particular surface, and then you pass your analyte on this, and if there is pesticide present, enzyme will be inhibited, you will not get any activity or the reduced activity. So, this is indication of the presence of the pesticide in your sample. So, with this I will come to the end of my talk, and I would like to conclude with the statement that, so I think you all must have realized by now that enzymes, they are important and indispensable tool in the hands of the biologist. The knowledge generated can be put to innumerable uses. So, you just have to your thinking is the limit, because you expand your thinking, you can get n number of applications of the enzymes, and the most recent one, it is the genome editing. So, you all know the Cas enzyme, which is an endonuclease which is making the cuts required for the genome editing. So, the sky is the limit, rather I would say even the sky is not the limit. So, just you need to have the expansion of the thinking. So, with this I thank you very much for your patient listening, and I would be happy to answer your questions if any. So, without question the talk is never completed. This is what I believe. So, either you have understood everything or you haven't understood anything. So, this is what it means.
They are doing an applied aspects of the enzyme, since they are not exposed to the basic. They may not be. I think many of you might have been working on some metabolic detoxification.